Hello, welcome back. My name is Jeff Martin. We're here at Advanced Therapies Week in Dallas, and I'm here with Rachel Legman from Repligen. Rachel, what is your role at Repligen?
My role is Senior Director of Technology, leading the cell and gene therapy at Repligen. I'm bringing more than 25 years of experience dealing with the new modalities, manufacturing, for bringing a better process into the market.
That's good. That's good. So what are the challenges the market faces today in viral vector processing? And what are the trends that you think are available currently to overcome these challenges?
So I'll have to focus on several challenges because this market is still facing multiple challenges. But since my expertise is more, and my company expertise is more in the bioprocessing, and we are having a lot of innovation gearing for these challenges, so I'll focus on the challenges that are relevant to how I can contribute. So the first number one, in my opinion, is the high drug cost. Due to the complexity of the product and the complexity of the process. It is a big red flag when there are approved drugs that are withdrawn from the market due to the high cost. So that has to raise a big challenge into the market. We have to do everything we can to approach these patients that are priceless to get this drug, but it has to be affordable to everybody in the globe. So this is, I would say, number one in my opinion. The number two, which is related, is the complexity of the process due to the complexity of the product. So with this relevant is that I'll give you a few examples on that. If I'll walk through the workflow, the host cell is not growing to high cell density. So how can you produce a lot of drug if the host cell is not growing to high cell density? Especially in the C-train and in production vessel. So we need to push this cell density high, and maybe I'll touch upon that later. The other workflow challenge, in my opinion, for the complexity of the process is including transient transfection. So transient transfection, if you work in manufacturing, this is not friendly. You have, especially when you go scalability. So you have to dump large complex, combine the right concentration of plasmids and the right concentration of the transfection reagent into the bioreactor, and you hope that you get an efficient transfection. And that's not happening when you go scalability. So this is something that we have to address very carefully. Another point, especially with the AAV market, that it is inside the cells. So you need to lyse the cells. I don't have to tell you how tall it is on the downstream people dealing with this nasty material that I would say. Another point that I want to make effort on the workflow is the community of the pharmaceutical people, scientists, like to work in their comfort zone, what they know about. So using legacy tools not necessarily is good for this market. You have to be open-minded and look for a better fit technology that address this specific market. So this is another challenge within the workflow. The fact that you have product that is basically cells and virus, you cannot do viral clearance at the end of the process. So you must integrate a risk mitigation strategy along the process in order to avoid the contamination and failing batch that will increase the cost overall. So this is something that you have to work around it. Another point that I think it's critical is to make the right decision during the process. Unfortunately, there is a lack of understanding of the product and the mechanism.
That's interesting.
And therefore, there are not enough analytical tools that you can integrate into the process. So by integrating process analytical tool, give you better data online, on time, in order to make knowledgeable decision how to move forward. And usually people are working in a black box. They're making decision based on indirect relevant parameter that they see. So this is something that we must integrate process analytical tool into the system in order to make the right decision. Another barrier that I'm seeing here is the lack of concise and detailed guideline from the regulatory, and this is in progress. People are feeding the regulatory learning from the pharmaceutical, and eventually it is working. Another barrier that I would say is the lack of resources and funding, and not less important, experienced people that has the ability to scale up and so this is lacking in this market, and we have to overcome them in order to reduce the overall cost.
Right. Yeah. So staying on overall costs, let's get into some specifics. What are the most exciting technologies that we can be leveraging to reduce these costs?
So if I'm touching upon better fit technology, you have to look for technology that design addressing for purpose the product that we are dealing with.
Yeah.
So let's focus on viral vector.
Yep.
So you want to intensify the cell density. I would highly recommend to integrate perfusion system. So when you are integrating perfusion system, you enhance the cell density both on the C-trend, that it's important to simplify the process, and in the production vessel. And you have to look for the right perfusion system. So this is one point. Integrated solution, once again, you have to look around the market and look what are online, reliable, and rapid analytical tool that will provide you more information about the critical attribute, quality of your product. Usually you have analytics that are indirectly give you the process parameter, but it's very basic.
Yeah.
So as we speak, the market is gearing to that, but it's still missing.
Right.
And you have to integrate the analytic into your process. So look for how you can do that the best way.
Right.
And as I said, this you have to build the risk mitigation. So you have to work with single use, so you don't have to do all the validation and reducing the cost. And you have to do closed system, and you have to work with automation in order to reduce the error from human. So there is
That's getting very big nowadays, by the way.
Exactly. And I can feel it because when you're on the floor and you have all this deviation and inconsistency from lot to lot, that's prevent you from moving into clinical. And also increasing the cost because you're doing cycle after cycle of development, and you need to reduce this number of cycle and move fast to the clinical to be successful at the right cost.
Right. So right now, gene delivery is dominated by AAV and lenti. Over the next 3 to 5 years, what other kind of viral vector forms do you see kind of entering into this space?
Well,
if any at all.
Is
there room for anything else?
There is definitely a room of improvement all the time. We have to continue improvement everything. Myself, as a bioprocessing tools that we are contribute to this market, we always improve the process. So to your point, I would say that now I'm less optimistic that within the next 5 years will change the AAV and lentivirus. We do need to improve the process. We do need to shrink the workflow and simplify that in order to reduce the cost. Because if you look at cell therapy, for example, the major component that enhance the cost of the process is lentivirus.
Right.
So you have retrovirus, lentivirus, any envelope virus that people are working on. So I don't see in the next 5 years, and maybe more, I was more optimistic 5 years ago, but now I'm more realistic.
Okay, that's good.
It's still a great solution for the patient to use this viral vector, and we see successfully things happening. I was part of the development of Zolgensma. I was lucky that this process came to me from academia, and I had to industrialize it and move it into the market. And within the 7 months, they got approved and bought by Novartis. So this is something that you always have to improve. But having said that, I still see new modalities emerging in
Yeah.
order to bypass the viral vector.
Okay.
Because this is a process, very complicated process. So if you are not improving your process by making stable cell lines, so you bypass the transient transfection.
Sure.
You make a capsid that will be secreted out so you don't have to lyse the cells. So all this improvement must be made. But the new modality that I'm very hopeful to merge up is the gene therapy that are using mRNA, which is a non-viral based therapeutic product. And it has its own complication, but it's easier to work with because you're not using biologic to produce it.
Right.
I believe also that adenovirus and other infectious virus in the oncolytic market, especially either for vaccination, immunovaccination, or vaccine, or whatever you want to deal with these viruses, it will be bypassing the transient transfection because this is infection. So you don't need a large and expensive plasmid. You don't rely on supply chain for the transfection agent. So this is a little bit more easy and simple process to use.
Right.
So I hope there will be non-viral effort moving like maybe 7-10 years from now, but still
for the time being,
or improve what you have and reach out to this patient that we have to deal with all this rare disease that there is no cure today.
Right.
And improve the life of people like hemophilia, and so this is working. And I met an exciting person that was getting the sickle cell. And it was just amazing talking to her and see how her life improved. So we always have to keep that in mind.
Exactly.
And develop everything that will reach the target.
So
now it's a viral vector. In the future it will be non-viral vector, but we have to be very cautious of our target and timeline.
That's excellent. So I've heard this phrase being thrown around a couple times at this conference: process analytical technologies. Okay, so what are those, and where do we see these real-time technologies playing a role in this space, and what are the results to date that are kind of increasing the optimism for these things?
Yeah, so that's very high level point is the integration of the analytical. As of today, currently, as I mentioned, we have only the basic analytical into understanding the process parameter, which is important, like pH, dissolved oxygen, CO2, temperature, everything that give you understanding if the cells is healthy, the viability of the cells and the concentration of the cells. That's give you a very good input, but nothing is on the quality of the product. And we must find and work gearing to build more sensitive, reliable online analytics that will address the product itself at each steps of the process. So at Repligen, for example, we are having a online analytical tool on the downstream side for protein for sure, but I want to focus on the AAV, plasmid, mRNA. This one we can analyze with our variable pathline technology that you integrate, and we're already doing that. We launch into the market because we are gearing for purpose in this case to improve the integration of analytical tool into our system. But this analytical tool can be integrated to any system, so we are not blocking the market from having a real knowledgeable decision how to move forward, how much of material you have, how clean the material, monitor and controlling with feedback control your process. So for example, I'll give you the AAV. So when we are integrating the analytical tool we have, the variable technology, into RSTFF system that we have, then you can know what is the concentration of your full AAV and how much of empty capsid you have. So when you know that during the final step before fill and finish, this is very powerful. This is a game changer. So now what we are doing, we are integrating this analytical tool into Chrome system, and to the TFF system, and we're helping other customers to integrate that into any
Whatever they need to.
skid they have.
Yeah.
So of course we would like them to use our skid because it is redesigned for purpose. So it has a low hold of volume, very fragile pump to handle, because when you're talking about this modality, it's not just the product and the consumable you're using for the separation and purification.
Sure.
It's also very important to look at it holistically and see the journey of the molecules.
Right.
If the molecule is on the journey from one spot to another is stress or hold too long, this modality, you're losing it.
Exactly.
Especially the potency, that's what we are looking for.
Yeah.
So that's how we are bringing our vision for purpose in our technology.
Nice. Well, it all sounds really good. Where can we find you today if this sounded interesting to somebody? Where can they get in touch with you?
Yeah, one of the powerful network that this conference is the app network. So everyone that wants to reach out, as they did already, please reach out to me to meet with me, and I will discuss in more depth and in more lengthy about specific needs they want for their specific modality, because we are using that for multiple modalities, and I have experience, and we have a team with a lot of experience and collaboration and understanding the weakness of what they don't know. Come over and ask us. And I can reach out to you as well. So this is, I would say, let's leverage from the network up.
Sure. So the app, Rachel Legman, and on LinkedIn. Are you on LinkedIn? We can find you on there.
I am on LinkedIn.
Okay, Rachel Legman.
I'm excited to answer a lot of questions that people already reach out to me during this conference.
Excellent. All right. Well, thank you very much for your time. I really appreciate it, and looking forward to the future for you guys.
Thank you, Jeff, very much.
No problem.